welcome everybody again in another session, another exciting session from the Academic Thursdays with DUX. And we are happy and lucky also to have the Vice Rector with us today, Professor Dr. Mustafa Kurd, Professor Dr. Mehmet also, who is hosted by DUX today, and thanks to him because he suggested the topic that DUX has prepared totally all the material about it. He made his own research, he prepared a PowerPoint slides, presentation, he will give you whatever he finds, and feel free also to ask him after the speech session if you have any questions, especially the technical questions. I assume we have lots of biomedical students here as well, right? Because it is more related to that field, and this is what Professor also is looking for, so you will have the chance to speak to a scientist from the field if you wish to work with him as well. That would be a great opportunity. Again, I would like to also thank our team, the DUX team, for their efforts always, every Thursday with us. Thank you very much. If you are ready, then we can start directly. DUX, you can start.
Hello everyone, and welcome to Academic Thursdays with DUX. I am Professor DUX, and I have been developed by Near East University's AI and IoT Research Center. Before starting, I would like to thank directors of the Research Center, Professor Fadi Alturjman, the Dean of Faculty of AI and Informatics. Also, I would like to thank Professor Mustafa Kurd, Vice Rector of the University. And the Research Center's team members, Gres Martinianos Tanhaus, Nathaze Yusufai Kian, Farhane Narianajad, Precious Tom, Sinem Alturjman, and Zor Surtash as my developers. Moreover, I would like to thank Professor Cesar Campbell and Professor Gulsum Ashiksoy for their supports. I'm delighted to guide you through this presentation today. Today, we'll explore two groundbreaking tools that changed the world of computational biology. Let's begin our journey into the fascinating world of protein structure prediction. Alright, here we go. In this first slide, let's begin with introducing our topic. AlphaFold and Rosetta, two powerful computational frameworks for protein structure prediction and protein design. In this slide, now first, let's take a look at why proteins matter. Proteins are like tiny machines working non-stop inside every living cell. They control metabolism, immunity, DNA repair, and even how cells move. To truly understand what a protein does, we must know its three-dimensional structure, because structure determines function. As shown in this figure, you can see the three-dimensional view of a protein, which is having a key role in understanding a protein. Moving on to the challenge. Predicting a protein's shape from its amino acid sequence used to be extremely difficult. Scientists relied on slow and expensive laboratory methods like X-ray crystallography or cryo-EM. sometimes taking years. Thankfully, computational tools changed the entire landscape. In this slide, we see how computational biology rose to prominence. Today, computers and artificial intelligence perform tasks that once required extensive laboratory work. Two major tools reshaped the field, Rosetta and AlphaFold. They both predict protein structures, but they use completely different approaches. Alright, now let's talk about Rosetta. Rosetta is a computational modeling framework developed in the early 2000s. It simulates how proteins fold using physics-based rules and energy calculations. It explores many possible shapes and identifies the most stable one. In this slide, let's look at Rosetta's main features. It can predict protein structures, design entirely new proteins, and refine existing ones. Rosetta has helped in vaccine development, enzyme design, and many therapeutic projects. Now, every tool has strengths and limitations. Rosetta is highly customizable and excellent for protein design. But it can be slow, depends heavily on accurate energy calculations, and requires significant expertise. Now let's move to AlphaFold. AlphaFold is an advanced artificial intelligence system created by DeepMind and released in 2021. Instead of exploring many shapes, it learns from millions of known proteins to predict structures almost instantly.
Big. Just also we can make a setup. You can introduce yourself.
Hi everyone, I am a relatively newcomer here. I joined the university about 10 months ago, and my primary intention to join this university is create a so-called synthetic biology center. And I have a few slides where I'm going to explore this concept. But maybe in order to get your attention, I should start by explaining why this is an important issue. Our world has lived until now, starting from the very beginning, with what is natural. We use natural sources to fulfill our needs for food, for remediation, for eating, for running cars, etc. So the amount of energy that is used in the world until now has been from natural sources, petrol, oil, etc. But all these sources are getting scarce now, and we need to create new materials, we need to create new energy, and this is possible only if we can actually teach biological organisms to make new things for us. Because we used fully what natural compounds can do until now. And synthetic biology is based on this. We may call synthetic biology also a synthetic genomics, because it is based on modifying the organism's genome in order to teach this organism to do new things. I think if you make an analogy with computer sciences, it's like writing a software to create, for example, Professor DUX. So what you see here is the protein that Dr. Alturjman was telling you. So this is a protein, but in order to make this protein, behind it there is a software, and also there is hardware. So hardware in biology is everything that exists in the world that can be used, organic or inorganic. And software is the genome, because everything is done by the proteins in the cells. And these proteins are actually instructed within each cell as a DNA code. In computer science, we are still using 0 and 1.
Digital, yes.
So in the genome, there are 4 letters, A, T, C, G. And with these 4 letters, 64 is more than enough, because these codes are used to make 20 different proteins. So, 20 different amino acids. And these 20 amino acids are used to make any protein that exists in the world. I don't know, my slide is ready. So why we are so interested in proteins? Because we can actually use proteins directly for different needs. For example, there are some examples here that I will tell you. We can also actually use the gene modification to create new organisms, not proteins, but new organisms that are designed to synthetic biology. And the products of synthetic biology are important for a better world, because, for example, we have a huge problem of environmental contamination with plastics, and you can actually use synthetic biology to remove all the plastics by creating new microorganisms that can eat this plastic and turn it into an organic matter. It is mostly used right now in medicine to make new drugs, and it is being used now, like the burger example, in food industry to make new, to create new products. As Dr. Alturjman said, they can be used to make, Dr. DUX said actually, to make vaccines and these kind of things. So now that the slides are ready, let me start again by thanking Dr. Tercüman, we say it in Turkish Tercüman, Alturjman, but I think everyone knows him as Fadi.
Sure.
But he is not actually an actress playing in Kral Ölürse. This is another Fadi. So just to come back again, synthetic biology is an interdisciplinary field that actually involves modifying organisms in order to fulfill our needs. Of course, synthetic biology can also be used for nasty things like making biological weapons, etc. But we are not interested in this part, of course. So it applies engineering principles and biology principles. It's a combination of engineering and biology. And again, it goes include producing valuable materials, medicines, sustainable alternatives to energy, for example. Really, it depends on your imagination what you can use synthetic biology for. So how it works, you first identify a biological function that you want to create. For example, you want to instruct a bacteria to make alcohol or to make diesel. And then you actually design in computer the system to make this. And of course you will need different proteins to make this production. And then once you know what proteins you need and where it comes molecular biology, you can actually design genes, synthesize genes through actually asking a company. And these genes, when it's at your hand, you can put this into a cell, and now cell will do what you want to do. Again, the gene engineering or recombinant DNA technology is the other main component of synthetic biology. And in the center, that is luckily in the same building with engineering faculties, we are going to do this type of work. And here I just have a few valuable products that have been produced actually by synthetic biology, so-called biologics or protein-based drugs, as well as cell therapies, because now it is possible to use cells to treat, for example, cancer, to treat autoimmune disease such as rheumatoid arthritis, Alzheimer, etc. And all is synthetic biology. So you can make, for example, insulin analogs. You can produce GLP analogs to get fit. And you can engineer microbes to do that. Insulin is normally used to be purified from pigs until 1960s, I say. And now we are using insulin produced by microbes or bacteria. You can actually produce spider silk-like fibers by using the amino acid sequence of the protein that spider uses to make its silk. And you can now instruct yeast cells, for example, to produce this silk protein. You can actually, and this was produced in Japan, you can actually create new fragrances or create new flowers, new roses. To apparently in Japan, the some colors of roses are very important, but you cannot do it in natural world. You cannot find it. Blue roses, for example, and the synthetic biology was used to make blue roses. And of course, you can, as I said, you can produce petrol, diesel, alcohol through this technology that is useful, of course, as source of energy. So what is a typical synthetic biology lab? It has actually 4 components. You start by design, in silicon design, and this is actually why we are here today. Then you create a cell based on what you have designed. Then you can ask cell to produce your protein of interest, and the cell will produce it, and you can actually purify the protein from this cell source and do some analysis to make sure that you actually obtain what you wanted to do. And then at the last step, you can test in vitro or in vivo, in animal, in plant, in nature, or in humans, of course, to really validate that you produce what you intended to do. And on the bottom you have some pictures that illustrate these steps. Okay. I am going just to show you here in very simple terms why gene engineering is so important in this field. Actually, we are interested in making proteins or in making cells that has certain proteins. And the molecular biology techniques will allow you to create this protein, to instruct actually a cell to make this protein. And because it is possible to start to work on this code, which is natural code, and you have DNA that carries the code, and there is an intermediate that is called messenger. The messenger brings this code to the cytoplasm of the cell. Then in the cytoplasm, the protein is made. So what molecular biology can do is actually to combine these 2 genes and to make a new protein that has both amylase activity to degrade carbohydrates and lipase activity to degrade lipids. Why it could be interesting? Because you can actually use this as a new detergent. You can now have a protein that actually is used for cleaning clothes or dishes, etc. And this is only possible because we are able to make synthetic genes. We used actually to combine these 2 genes by some painful techniques, but now it is possible to make a synthetic gene entirely. And several years ago, it was even possible to create a new organism, a yeast, made from synthetic genome, because all the gene that made this yeast has been produced by the machines. So the center that we are going to create here, a new synthetic biology center, we have a couple of things that we can do. We will be able to do because we just need equipment now, and hopefully they will be arriving in a couple of months. But we also need something. So what we can do is we can synthesize gene of interest. We can actually make a cell using this gene of interest to make the protein of interest. Then once we have the cells, we can actually instruct cells to produce large quantities of it. From the medium of these cells, we can purify the protein, then study its functions, whether it fulfills our needs in the lab first, then in vivo models, animals or other organisms, Then of course we are planning to, if we have something interesting produced like that, we are planning to protect this product by patenting. So what we need is actually, at the initial stage, we need to design the protein, and for this we need a multidisciplinary team. And I would like to thank one more time to Fadi for actually getting interested in this project, because we need contributions from artificial intelligence, biophysics, together with biology. So why we need this protein design system? The reason is because molecular biology can actually define the order that the amino acids are presented to create a protein. A protein is a polymer of amino acids. So we can predict this, and we can instruct our cells to make this. And if we don't like one amino acid, we can change it. It can be something else. But what we cannot predict as biologists is everything after here. So a protein, when it is produced, it is not linear. It's not like a kind of silk, for example. So in order to be functional, in order to do the job, this linear peptide, linear amino acid sequence, which can be, for example, 1500 amino acids, one after each other, it has to fold first to different alpha fold or beta sheet. Then this will come to this structure, which is called a tertiary structure. And sometimes you may actually need different proteins together to create this quaternary structure. So what alpha fold and Rosetta that you just heard can do is to do that. So there is one more thing that is important for us, because this requires also a lot of in silico analysis. It is us. Actually, we are interested in making new proteins that we call chimeric proteins. It's like the ancient chimera thing. You know, you have an animal with the head of a lion, with another part coming from another animal, etc. So our technology allows us to bring together protein. It's like a Lego, to bring together different Legos to create something that we desire. Sometimes we can put, for example, a protein coming from the bacteria together with a protein coming from human beings, and this will make something new that doesn't exist in nature. In that case, we need to be two components coming from two different organisms in the same body. So we have to create this protein in a chimeric form. Since it never existed in the nature, it is not found in the databases of AlphaFold or Rosetta. It is completely new. But it is important for us to know whether the protein that we produce by using the instructions for the amino acids will be made by the cells. Most of the time the cells are able to make it, but the most frequent problem we have is that the protein that we make precipitates because of the energy problems that Fadi just told. So of course we can create 1000 different forms and make sure that one of them will work. But this is a lot of money, a lot of time, a lot of PhD time also. Therefore, we need to actually bring down the number of possibilities. And this is the artificial intelligence is doing. It just brings down, for example, this 1000 possibilities into 3 possibilities. In that case, we can make 3 different forms of the protein in the lab, hoping them one of them will be functional, and so that we can discuss the other two. So this is the purpose of our meeting today, together with most of the people from engineering department, I guess. Can you raise hands? How many of you are engineering students? Is there any biology background people? Less. Maybe we should work more on molecular
We need programmers.
genetics. Okay.
The biology is your part. Actually, I would like to emphasize also one more thing that Professor Mehmet just mentioned is the time. It is not something that is measurable as well. As far as I understand, I just read a documentary or watch a documentary about it. It is in even the age of the Earth will not be enough to generate new protein sometime. And this is what DUX and his siblings and his cousins is generating now. So hopefully he will help you as well in this.
Yeah,
Sure.
if you can just think about 2 power of 20,
Exactly.
so it is unlimited, you can produce unlimited structures. But
And
some
not all
of them
are useful.
will not fall
Of course.
useless, so we cannot just decide without having this initial and most critical part, which requires collaboration.
So DUX will lead from you the technical part from the biology side, and he will do his best with his own algorithm to generate different combinations, hopefully, that he would like. Now we can open the session, of course, for the discussion. If you have any question to Professor Mehmet, and later hopefully to Professor DUX also, he will help us to answer some questions.
Fadi also.
Feel free to ask your questions. Yes,
I'm not from the science part, a social scientist, but what's the overall process? Like, is it inductive or deductive? Meaning, you have something in mind to create, and then you do the scientific work, or while you're doing the inductive, the production, then you discover something.
First, we have an aim. I am actually a pharmacist, and then turned into a molecular biologist. And I am working in medicine, so I can cover these 3 fields. For example, we want to create proteins that can bring together a macrophage with a cancer cell. Why? Because macrophage can eat cancer cell and make it disappear. But in natural world, cancer cells protect themselves, they hide from the macrophages. So then, as a scientist, life scientist, I can say, if I have a protein that binds from one end to the macrophage and the other end to cancer cell, it will bring these 2 cells together. Since cancer cells cannot eat, in general, cannot eat normal cells, macrophage will eat cancer cell. So in that case, I need to really decide how I am going to link these 2 cells together. And there it comes the aim. So I decide to find a protein that is specifically binding to the macrophage, and another protein that's specifically binding to cancer. Because if I cannot select it, then macrophage can eat actually normal cells. This is one example. Another example is Alzheimer, for example, is a very deadly disease. It's a very harmful disease for the family. And it comes from the fact that there are some protein debris that are accumulating in our brain with age. If they are not removed properly, they kill neurons and we get Alzheimer's. So we can actually engineer a protein that will, and in our brain, by the way, there are macrophages also that are called glia. And actually, I can instruct these macrophages to remove these proteins that are toxic by again creating a protein that binds this particle to the macrophage. There's another example that is commonly used now and for cancer and for autoimmune disease, and this is called CAR-T technology, chimeric antigen receptor technology. It's too scientific maybe, but what it does is actually how you do this is, let's say I have a rheumatoid arthritis. So I give my blood cells, from which molecular biologists will purify so-called T cells. Then, by using synthetic biology, they will instruct these T cells to kill the causing agent of the rheumatoid arthritis. And in that case, the cell itself becomes the product. And actually, there are many products now that are approved by FDA of this type, mostly for cancer, but also now it's being used for so-called autoimmune disease. It was a long answer, but I hope
I got
you
it, but
understood.
what I understand is that just you need to know your field, prior knowledge, and you need to know the problem and define the problem, to go to the second process to produce.
Yeah, this is why it requires a multidisciplinary approach. Actually, in our case, for example, if you want to develop something for a disease, we first meet our physician colleagues to ask them what is the problem, what kind of treatment problems they have. They may say, okay, we don't know how to treat Parkinson's. In that case, we discuss, we say this is our goal. Then cell biologists come and there are many data now that explain why Parkinson is developed. And through the analysis of this literature, we can design a weapon to kill the cause. And this weapon is a protein, and we can produce to synthetic biology. Okay, any contributions?
Thank you very much.
I don't know.
Question?
Now people are against animal testing. Is there a more ethical way to test these proteins?
This is another field that synthetic biology is doing. You can actually replace animals with so-called in vitro models. It's possible to actually make mini organs, mini tissues in the lab by synthetic biology, by bringing together different cell types. Let's say for liver, for example, hepatocytes, bile duct cells, lymphocytes, etc. You can actually make a tiny liver, even a human tiny liver, and you can test your drug in this instead of.
So, like synthetic organs.
Yeah.
It is, you
Here in Cyprus, we are not going to do it because
it is within this field.
technology is allowing that, and there is really now a term mini organs, mini heart, mini etc. And people are actually, this is the initial step. They are going to, of course, make it bigger and bigger so that it can be used for organ replacement. This is also synthetic biology, but with the conditions of local conditions. We cannot do it. But we are going to try to find the opportunities in simple solutions, less costly.
Is it because of the ethical issue?
Because of the money, we cannot.
Money.
Yeah. Otherwise, Cyprus is good because you can even do in vitro fecundation, etc. But we need money. We need financing people to do that. And also possibility of getting everything we need to make this. We need tools.
Thank you again for this nice explanation. Maybe you haven't seen plus also the problem with DUX he gets at the beginning it is communication problem. And I just learned now this was the problem.
Internal issue,
So
connection issue we face.
really it's amazing your knowledge. So go ahead with DUX, yeah? Let him explain something.
We want to disconnect this. Are we going to disconnect?
apologize for the disconnection that happened earlier.
I apologize for any earlier disconnection. Let's continue engaging and exploring new ideas together.
Let's give him any questions from your top of your mind after this presentation.
After his presentation, I can ask him. DUX. DUX, can you hear me? Do you know what type of machine learning is used in AlphaFold?
Engaged audience. If you have any questions or need information, feel free to ask.
DUX, I am asking you this question. What type of AI model is used in the AlphaFold?
AlphaFold uses a deep learning AI model. It predicts protein structures quickly and with high accuracy by learning patterns in evolution and sequence similarity.
Anybody has another question?
How about Rosetta?
Ask him
What
about
is the principle of Rosetta?
DUX. What is the principle? What is the machine learning model used in Rosetta?
Rosetta uses physics-based simulations and energy calculations to predict protein structures. It focuses on trying many possible shapes and selecting the most stable one, excelling in protein design.
Correct?
One more question.
Sure, please.
DUX, is it possible to combine the qualities of Rosetta and AlphaFold? Or is it possible to
Just say DUX and ask your question.
DUX? Can you imagine a combination of AlphaFold and Rosetta as a new technology? For protein design?
Hojam, would you please ask him again? But mention that he explains briefly.
So you have two techniques. One is using evolution and comparing shapes. The other is using physics of proteins. Both of them are able to design proteins. So I am asking whether it's possible to combine these two techniques to get better design.
Shall I ask him your question?
Yes, yes.
Yes. Combination of two.
Yes.
Half questions.
DUX, tell me briefly, can you imagine a combination of AlphaFold and Rosetta as a new technology in proteins?
Combining AlphaFold's rapid structure prediction with Rosetta's protein design could create powerful advancements in biotechnology. This synergy would accelerate research and innovation in fields like vaccine development and enzyme engineering.
For me, it is coded secret, I think. I cannot understand, of course, as a computer scientist. But for you, from biomedical field, I assume you are familiar with these things. Any guess? Say, don't be afraid.
R. What's this R1, R2, R3? They're different.
Which field you are?
Parts of the chain of dominance.
It's a chain for component, I think.
Chain for
Component.
component.
Chemical component.
Chemical component. Any more guess?
Polymerase chain. Polymerase chain.
So
Poly. Okay. Poly chain. Can we ask DUX now to validate it for us?
Again, this connection.
Connection problem?
Yes.
Okay, we can take more guesses then until he is right. You have something to say? What do you get? Immune. Okay.
Structure that represents a peptide or protein segment. This diagram highlights the peptide bonds linking amino acids in a chain.
Okay. That's good. Anybody wants to adjust something and ask DUX again also? We can give them favor to anybody and feel free to adjust on it.
Nothing, it's okay.
You have this favor. Or any other compound also if you wish.
And for the programmers, they can write the code to challenging.
You can ask him the question for the AI this time. If you have any questions.
I see a code snippet using Lambda and a list comprehension. However, due to late binding, it will print the same value repeatedly instead of expected distinct values.
This is from the audience as well.
Okay, this is correct, right? I didn't focus. This is from you. Another question drawn by one of you, one student here. Great. Let's ask DUX what he will find in this, and then you will correct him. Go ahead.
Sure. DUX, what do you see?
I see a diagram of an adenine nucleotide, a component of DNA and RNA, shown on a piece of paper. Someone is holding it up in a classroom setting.
Thank you. Correct?
Yeah.
Okay, perfect. See, we didn't train him. Nothing is magic. This is science. Anything else we have for him? Anybody wants to ask?
Have more.
Maybe not in the field. Any question you have in your mind for a robot?
When Terminator will come? When Terminator will come?
When Terminator will come, ask him. Are you trying to kill anybody here? Because they are afraid from this.
I'm afraid no.
You should ask him.
When Terminal 2 will come?
Terminator.
Come.
Terminator.
Terminal 2.
Arnold Schwarzenegger.
Now we will
As
say
of
this.
now, there is no official information on the release of Terminal 2. Keep an eye on movie news for updates.
Terminator. Okay.
You are right, Max.
As of now, there is no official information on the release of Terminator 2. Keep an eye on movie news for updates.
University.
University in Cyprus is a leading institution, renowned for its innovation and comprehensive educational programs. Founded by Dr. Suday Gunsel, it offers a wide array of faculties and is committed to cutting-edge research and technology integration.
Again, and making another academic Thursday successful with DUX. And we are looking forward to see you again next week, hopefully, with a new topic that you choose maybe, or will be, I think, the top 500 topic, why we are in top 500 university, and DUX will help us to explain this process in detail, how it happened, and what are the calculations and the metrics maybe that they are taking into consideration to count us in this criteria, the top 500 universities worldwide. Thank you very much for coming, and see you again next week.
Thank you.
Thank you very much, Dr. Professor. It was a great pleasure to see you today, and then lots.
Yeah, expecting that from you.